Hello, and thank you for coming. Okay, without further ado, let me start. So, negative stimuli get our attention. Whether a stimulus is good or bad is sort of the earliest and automatic determination that we make about a stimulus. But our reactions to various types of negative stimuli differ depending on the stimulus itself, its contents, and even our internal state. So, for example, we recoil from some negative stimuli, but are attracted to other negative stimuli. So, some years back, we decided to figure out what is it that actually attracts people to some stimuli and makes them withdraw from other stimuli. So we collected, actually, no, let me back up just a second. So let me talk about the threat dimensions. So the first threat dimension that we identified is agency. So basically who or what is about to attack you or is threatening you. So it could be a human, could be an animal, or could be some inanimate thing like fire or flood or what have you. Another very important dimension is capability. So the amount of harm that this agent can inflict on you. So it could be deadly, like someone with a gun, could be high amount of harm, like this tough-looking dude there, or, a minimal amount of harm. Like this baby's very angry, but he's probably not going to do, you a lot of harm. So another critical spatial dimension, and one that we'll be testing explicitly in our studies that I'm going to present, is the spatial dimension. So towards whom is the threat oriented? So most critically, when it is oriented towards you or me, it is, as you'll see, it produces very quick responses and sort of a lot of activation of the subcortical system. When it's produced towards others, like in this instance, the reaction can be either that of fleeing or helping or approach, and also we have a situation when it can be towards self. Another dimension is the temporal dimension. So when is the threat occurring? Is it occurring right now? Is it occurring in the near future? Or has it already occurred in the past? So these are not the only threat dimensions. There's also proximity and various other things, but I'm going to skip over that. And the basic idea is that you take all this information from the stimulus and you basically compute your most appropriate response in that situation given the context and given your internal state, which could be to flee, to fight, to learn from the situation, or to laugh about it if it's ridiculous. So to examine how we process these things, we started by collecting over 500 scenes from the internet. This work was largely done by my Arraean postdoc, Jasmine Boshayan. So the scenes were subdivided into these 4 categories initially. So the direct threat is obviously someone attacking you right now, basically to your face. Indirect threat is someone else attacking the agent attacking someone else. Threat aftermath is where the threat has already occurred and is in the past, and non-threat stimuli are sort of the neutral condition, if you will. So you'll note that the direct and indirect threat differ in the spatial dimension, but are the same in the temporal dimension. The threat is occurring right now, but towards someone else versus towards you. And the indirect threat and the threat aftermath differ in the temporal direction. the threat is towards someone else, but in one instance it's already occurred, and in the other it is already in the past. And obviously this is not a fully balanced design because it's very difficult to come up with pictures where you yourself are hurt and dead, right? So anyway, to do this, we asked 3 groups of subjects to rate the scenes. As soon as I get this to click through, and asked them the following questions. So the first group was asked a question that was designed to elicit, to basically rate, to identify the direct threat scenes. So the question was, how much harm might you be about to suffer in the scene if that was your view of the scene? We used the Likert scale with these anchor points. The second group was asked a question to identify the indirect threat scenes. So the question was, how much harm might someone else, but not you, be about to suffer? And the third was designed to identify the threat aftermath scene. So how much harm might someone else have already suffered in the scene? So what did we get? So the first group responded with highly distinct ratings for each of the scenes that basically went, from the highest to lowest for direct threat to indirect threat to negative, and to neutral scenes. So the second group likewise gave the highest rating to the indirect threat scenes, but also gave fairly high ratings to the direct threat scenes, and the subjects explained it as there being a possibility that whoever was attacking you, attacking someone else, could also attack you in that scene. So that's why this was the smallest difference. These were all highly significant differences between each of the threat conditions. And the third group predictably rated the negative negative threat aftermath scenes most highly. Okay, so let me just. Okay, so then we looked at the speed of threat detection by having subjects do a reaction time task in which they had to identify whether the scene had a threat object or not as quickly as possible. And for direct threat scenes, so these are the the conditions in which there is a weapon present or not. So for the direct threat scenes, we had the fastest responses by far, then indirect threat, and then the threat aftermath were the longest latencies, which we did not predict at all. We thought that the neutral condition would receive the longest latencies, but because it had no threat in that. But in fact, this RT pattern is present in every study that we run, including in eye tracking, fMRI, MAG, what have you, with these scenes, with line drawings. This is highly consistent and replicable. Right, then what's happening in the brain? This is a contrast where each of the conditions is contrasted against the neutral condition. So the colors match of the activations match our condition colors, right? So as you can see, these conditions activate sort of a related but also distinct set of regions, and the direct threat condition actually doesn't activate that much cortex. As you'll see, it tends to activate some of the subcortical regions more, whereas the threat aftermath condition activates most cortex of any of the conditions. So looking at some of the subcortical regions, in the periaqueductal gray, we have sharp peaks of responses to the direct and indirect threat conditions. And of course, periaqueductal gray is associated with threat proximity evaluation and has anti-nociceptive properties. Likewise, the amygdala, which of course does a lot of different things, also reacts highly initially to the direct threat images, but then followed by indirect threat, and later on by mere negative or threat aftermath images. And the orbital and then ventrolateral cortex are sort of a mixed bag. They respond to all conditions. Of course, this is a large region that does a lot of different things. Now, looking at the regions that are activated most highly to the threat aftermath, we have a set of regions, perihippocampal, retrosplenial, and medial prefrontal cortices, that have been identified in several different tasks, or actually task-free mentalizing. So one of the things that this set of regions consistently pops up in is contextual associations, as work by Alyssa Aminoff and Moshe Bar and myself has shown. This set of regions activates every time you have to process strong contextual associations, and has a nice temporal progression and activations shown by MEG from perihippocampal to retrosplenial to medial prefrontal. Okay, so we looked at also MEG dynamics during the same task, and here I'm comparing the direct threat versus the merely negative or threat aftermath images. So the red here is the activity that is stronger for the direct threat images. So from, I don't know if you can see the labels, but from visual cortex to perihippocampal cortex, we have strong activity going on for direct threat images, mostly in the theta and low alpha band. That continues at about 200 milliseconds for visual and paramygdaloid cortex activation, and also for perihippocampal connections with ventrolateral prefrontal and ventromedial prefrontal cortices. Okay, so another study that we did was looking at lateralization and threat perception. So what we did was we took the scenes and put them in pairs, presented in the left and right visual field, where the pairs were either direct threat versus a neutral scene from the match context, and we matched them as well as we could, or the threat, merely negative threat aftermath scene versus a neutral scene from the same context. So, and the task was to simply respond to the side containing a threatening or negative image as quickly and accurately as possible. We also, so in one of the studies we did eye tracking, and also did an MEG study. So first, the eye tracking. So there's what we've discovered. So I'm going to talk about the saccadic data first. So what we found was that the initial saccade velocities to identify, we didn't tell the subjects to look at any particular scene. They chose to do it freely themselves. So they first looked at the threat scene on the left, and that was faster by a significant amount than looking at the threat aftermath scene. And it was sort of reversed, although not quite significant, in the right visual field. The biggest effect was in dwell time, or how much time they spent looking around the scene, how many saccades and how much fixation was there on the scene. So they fixated much more on the left visual field and significantly more on the threat aftermath scenes versus direct threat scenes, which are, of course, more complex. And for the manual responses, we had huge effects of visual field. They're being faster to respond to the left visual field than to the right visual field, and also a huge effect of condition. Subjects being faster to respond to the direct threat scenes versus merely negative scenes. Now, as far as the MEG activity, we extract activity from periamygdaloid cortex label, shown here, from each hemisphere. And basically, the upshot is that the right hemisphere amygdala, or amygdaloid complex, whatever you want to call it, responds strongly to both threat and negative scenes at about 200 milliseconds. So there's a sharp peak, but it doesn't really differentiate that much between the conditions, whereas the left periamygdaloid cortex does not have such a sharp response initially, but then starts differentiating significantly between threat-negative scenes at around 400 milliseconds. Okay, so another thing that we wanted to explore was how much visual information do we actually need to make this threat prediction. Obviously, these scenes are they differ in color, and amount of detail, viewpoint, and so on, right? So what we wanted to do is to sort of clean that up and just provide the basic visual information and see if people can do that. So Jasmine here painstakingly traced all the threat scenes and made them into line drawings that are, while removing some of the background details. We then did the same rating study as I showed you initially, and discovered that the ratings actually match very closely between the photo scenes and the line drawings. In fact, almost perfectly. We did clustering of the results, and then popped a new category that we call deadly threat, which was basically all armed hostile men, and no animals or no unarmed individuals. It was all basically men pointing guns at you. So that was the new category. But I'll continue with the 4 categories for the time being for the next few slides. One important consideration for making these line drawing scenes was to be able to bias stimuli towards the magnocellular and parvocellular pathways more easily to study their contributions to threat prediction. So about 10 years ago, okay, let me go over quickly the qualities of these pathways. So as you probably know, the magno pathway has high temporal sensitivity, is color insensitive, sort of coarse, low spatial frequency, and can pick up very low luminance contrast. So our stimuli had less than 4% luminance contrast, and they looked like this in the original study. This was done with objects initially, 10 years ago. So the parvocellular characteristics are color opponency, of course, for red and green, fine detail, but needs high luminance contrast to respond. And we did heterochromatic flicker photometry to find the isoluminance point for each subject, and use stimuli like that. So in this study, what we showed was even though the magno objects were objectively and subjectively less visible, subjects actually responded almost 100 milliseconds faster to identify the object in the line drawing. And what we found was that with the parvo bias stimuli activated the ventral stream, shown in blue, and the magno bias stimuli activated mostly the orbitofrontal cortex and anterior temporal lobe, and also some dorsal stream areas, and the amygdala, the right amygdala actually, which is not shown here. We did dynamic causal modeling, just briefly, model 4, which predicted that basically there's a feed-forward drive from basically visual cortex to the fusiform gyrus, and a projection to the orbitofrontal cortex with a feedback connection to the fusiform gyrus was selected as the best model. And we had basically evaluating the contributions of the parvo and magno pathways. We found significant increase in information passing from MOG to fusiform gyrus for the parvo condition in the feed forward, and to OFC and back to fusiform in a magno condition. And I am kind of running out of time, I guess. So we did the same thing for our scenes. As you can see, the magno scenes are extremely hard to see, but as subjects were actually able to do just fine with them with RTs comparable to the Parvo condition, here shown in the middle in the right panels. Again, the same RT pattern as for the photoscenes. And so the interesting point here is that the direct threat, if you take unbiased scenes and take only the direct threat versus the negative aftermath scenes, you get this activation pattern that basically ventral stream activation, mostly parahippocampal cortex for the merely negative scenes, and prefrontal activation for direct threat. And when you do a Magno versus Parvo scene comparison across all conditions, you get a very similar pattern. Again, a ventral sort of prefrontal dorsal split. When you do the overlap of these conditions, you find that there's a lot of overlap between direct threat and Magno, and threat aftermath or ambiguity and Parvo. Also, comparing the line drawings to the photographs, you see a lot of overlap as well. So as I'm out of time, I'm going to skip over the Meg dynamics and the phase stuff and just summarize the results up to this point. So threat prediction from line drawings is similar to that of color photo images. And the findings replicate well with line drawings. We had a different task, different scanner, 3 versus 1.5, different cohort, pathway biasing. Despite that, they replicate very well. Threat stimuli activate orbitofrontal regions that overlap with those activated by magno-biased stimuli of any type. And conversely, the merely negative threat aftermath images activate ventral occipital regions that overlap with the p-biased stimuli activations. So the M pathway may have a predictive bias for clear threat and reflexive action versus the P pathway, which has probably a bias for reflective analysis and threat ambiguity images. Thank you. And let me just scroll quickly to my acknowledgement slide. Okay, so I want to thank people in my lab, my collaborators, and the funding bodies. Thank you.